So we are doing a little mini lecture. Still in chapter 2, but this is on protein conformation. And what that basically means is the forming and shaping of a protein. So remember, we went through transcription and translation of proteins, which is basically putting the amino acids together. And then the amino acids are going to fold a couple of times into its final shape or conformation. So proteins, which account for about 50% of the organic material in the body, or 70% of the body weight, and I guess that depends on how muscular you are. But proteins play a role in almost every physiological process. So again, if you think about proteins, they can be structural, they can be chemical messengers, and chemical messengers like hormones or neurotransmitters. So proteins can also be enzymes that drive reactions, right? So they would be like biological catalysts that drive metabolism or any kind of chemical reaction. So not just structural, but there are other many other things that we use proteins for. And proteins really are, again, they're organic and they contain carbon, hydrogen, oxygen, nitrogen, and sulfur. So this is sulfur right there. We went through this in biomolecules, but again, proteins are polymers or repeated subunits. So polymer just means it's a repeated subunit of various different amino acids. So this could be various and different amino acids. So there's 20 different kinds of amino acids. And different proteins are different combinations of these 20 amino acids strung together or chained together with peptide bonds. So quick review, an amino acid is going to contain, they all contain an amino group, a hydrogen, a carboxyl group, all bound to a central carbon. And then what makes each one of those amino acids different is the R group. So this is going to be different on each of 20 amino acids. So that R group, it's also kind of known as the functional group. Is going to be different. So alanine, you have a different R group, right? So that's the R group. This is the R group on tyrosine. This is the R group on glutamate. And again, that's the R group there, right? So they're all different. This plays a role in how the protein folds or conforms. So that is what conformation is. Again, a little bit more review. Peptide bonds are formed between the carboxyl group on one amino acid and the amino group on another amino acid. And so we're pulling out a hydroxyl from the carboxyl group and a hydrogen from the amino group, and we are forming water. So this makes this a dehydration or condensation reaction. RXN is reaction. And you're forming that peptide bond between the carbon and the nitrogen, right? So the carboxyl group has a carbon, amino group has a nitrogen. You're forming a bond there that will become the peptide bond. And so these peptide bonds are joining basically amino acids in a string. Now, if you have less than 50 amino acids, it's called a peptide. If you have more than 50 amino acids, I'm getting sloppy, it equals a protein. So that's just nomenclature. So again, the monomers or subunits of proteins are the amino acids. And each one of these amino acids have different side chains, which is the R group, or also known as functional groups. So a lot of different names, right, for those. Some are polar, some are nonpolar, some are ionized, right? They're positive or negative. They have one or more electrons or one or less electrons. The human body can only synthesize some of these amino acids. Others are needed to be obtained in food or through your diet. And those are the ones that we call the essential amino acids. These essential amino acids, they all come in animal proteins. You have all the essential amino acids in animal proteins, but in plant proteins, no plant protein has all of the essential amino acids. And so that's why vegetarians and vegans have to combine correct plant proteins so they can make sure they're getting all of the 20 essential amino acids. Down here is kind of a looking at the R group or side chain or functional group. And you can see nonpolar, all the bonds are shared equally. Polar, electrons are not shared equally. Ionized, you're going to have an ion or an ionized molecule on it. Now, proteins in nature don't appear like a linear string of beads. And this linear string of beads is akin to just a string of amino acids. Those are the beads, right? Beads are the amino acids. What happens is as these amino acids get connected together with peptide bonds, they start to have interactions between the side groups and as well as the hydrogens. So I would say between the hydrogens and then the side groups. And so this makes the protein kind of bend, twist, and fold into a more compact structure. And the final shape is called its conformation. So a protein's conformation is its final shape due to all of the bending, folding, twisting. And so levels of protein structure or conformation are the primary structure, the secondary structure, the tertiary structure, and the quaternary. So if we're looking at the primary structure kind of looks like a string of beads, right? So they have the beads, and that is really a string of amino acids, right? So string of amino acids, these are all peptide bonds here between the different amino acids. That's the primary structure. But remember, each one of these amino acids are going to have a hydrogen somewhere on them. And those hydrogens are going to be attracted to the oxygen or the nitrogen or one of the other components of the amino acid next to it. And so these hydrogens are going to fold or cause a folding. So the hydrogens cause folding into the protein's secondary structure. And that really only takes on two different forms. It'll take on one being the alpha helix, where it like curls around itself, or a beta pleated sheet where it just folds back and forth. And that's due to the hydrogen bonds. And so you can see the hydrogens are kind of attracted to the oxygens here, or if you're looking at the alpha helix, you can see the hydrogens being attracted to the oxygens here. And so that is the secondary structure. So due to hydrogen bonding. Now the third or tertiary structure is going to be due to the R groups, also called side chains or functional groups, same thing. Remember, they're different on every amino acid, and so that's really going to determine the tertiary structure or final conformation of the protein. So let's go through this step by step. The primary structure, again, there are two things that determine a primary structure. The first is going to be how many amino acids are in the chain, right? So number of. Another way to say it is how many amino acids in protein chain or peptide chain, depending. And then number two, the specific types of amino acids at each position along the chain, right? So you're going to have different amino acids. Sometimes they're the same ones right next to each other, and sometimes they're different. And there's thousands of different combinations. But that's going to make up the primary structure. And so a polypeptide in the primary protein structure is analogous to a linear string of beads, each bead representing one amino acid. So that's the primary protein structure. Now, the secondary structure or protein structure is due to hydrogen bonding. And it's hydrogen bonding between the hydrogen of one amino acid and the carboxyl group of another amino acid. And so the common structures are going to be an alpha helix. So this is just the Greek letter alpha, or a beta pleated sheet, right? So this is supposed to be a beta, and that's supposed to be an alpha, and alpha looks like this, and beta pleated sheet is more folded. So that is the secondary structure. Tertiary structure, protein structure, the third folding, if you will, is due to the bends and loops in the chain due to the interactions between the R groups, also known as the functional groups, also known as the side chains. How did they get so many names? I don't know. And so you're going to have all kinds of different folding due to the different properties of the different R groups of each amino acid. So the order of the amino acids is really important because the order of each amino acid in the primary structure will determine the tertiary structure. So the tertiary structure, it's the associations between the amino acid side chains. These interactions fold the polypeptide, meaning many amino acids, into its final three-dimensional conformation, making it a functional protein. Now, if this tertiary structure is broken, it's called denaturing. Disrupted or broken due to heat, right? Extreme heat can denature. Really acidic environment can denature. And when a protein denatures, it becomes non-functional. So that's why extreme heat can be very difficult. It renders enzymes non-functional or chemical messengers non-functional. And so these bonds are due to different factors depending on which amino acid is next to which amino acid. So you can have hydrogen bonding between the amino acids next to one another. You can have ionic bonding, right? So you have a cation and an anion. You could have hydrophobic interactions, meaning they repel one another. You can have a covalent disulfur or disulfide bond formed, and you can have what are called van der Waals forces. They're just really weak interactions, so it's forming this bend right here. It can be a combination of any. You can have two hydrogen bonding factors next to one another, and then you can have disulfur bond forming. So it just really depends on what the side chains are. Quaternary protein structure, that's when you have more than one polypeptide chain. So you have to have an example. Hemoglobin has 4 chains, right? You're going to have one chain, you're going to have 2 chains, you're going to have 3 chains, you're going to have 4 chains. Each one of these chains is going to have a hemoglobin molecule in it. And so it's really 4 tertiary protein structures combining together, and that forms the quaternary structure. So our learning objectives here, you need to be able to describe the primary, secondary, and tertiary structure of a protein and what creates it or is responsible for each of the formations, right? So what's causing that particular folding? And then what is the final shape of a protein referred to as? And so I want you to answer those 2 things. Why is this important to learn? Well, an example of a protein not being formed correctly because of a single amino acid being incorrect in the primary chain. And an example of that is sickle cell anemia. So in sickle cell anemia, during transcription and translation, of making the protein, right? And the nucleus of the cell, the messenger RNA codes for one incorrect amino acid in the chain. And so this is a molecule of valine, an amino acid of valine replaces what should be a glutamic acid. And so it's just one incorrect amino acid, but what happens is the protein doesn't have the right shape. It's missing an exact shape. So if you look at this hemoglobin right there, and you look at this one's folded just slightly different, but enough where the hemoglobin molecules that would go here are not structured correctly. They will bind oxygen, but they can unfold really easily. And so people with sickle cell anemia have a decreased ability to carry oxygen, right? And so they have low oxygen levels. And they can sickle or go into a crescent shape really easily. And this is just due to one wrong amino acid. So it just doesn't fold or conform 100% correctly. So just a quick review. Conformation, so a protein conformation is the final shape of the protein. And it starts with the primary protein structure, which is the number of amino acids in the chain. And number 2, the specific type of amino acids that are positioned next to one another along the chain. After the primary protein structure, you're going to have a secondary structure form due to hydrogen bonding. And remember, it's bonding between the hydrogen of one amino acid and the carboxyl oxygen of another amino acid. And so it's either going to be an alpha helix or a beta pleated sheet. Then the secondary structure allows the tertiary structure to form, and this is gonna be due to the interactions of the R groups or side chains, and it can be hydrogen bonding, ionic bonding, attractions, um, covalent disulfur bonds, van der Waals forces, and then if there are more than one chain of proteins together, you can have a quaternary or multimeric protein structure form. And that's it.